AP 120, Lab 7. Topics: diffusion across a membrane. So, here's a few terms that I want to make sure you understand. First off, solution. Solution is when you have one or more substances dissolved in another substance. So something dissolved in something else. Usually we're talking about liquids here. All right, then you have the solvent. The solvent is the substance doing the dissolving. And what is always the solvent in our class? Hope you said water. And then the solute. The solute is the substance being dissolved. So we look over here, we have a glucose solution, 1% glucose. Glucose is the solute, water is the solvent. If you have 1% glucose, then the rest has to be 99% water because you end up at the end with 100%. All right, so here's some examples. Solution A, solution B, C, D. What is the solvent for solution A? Solute, percent water. Again, solvent, solute, percent water for B, for C, and for D. Feel free to pause the video to figure out the answers. And it turns out solution A, solvent is water, solute is glucose, and there's 90% water. Solution B, the solvent is water, the solute is sodium chloride, and there's 95% water. Solution C, the solvent is water, the solute is protein, and there is 89% water. Solution D is a trick. It's not a solution, it's just water. All right, here is a lovely cell. And cells have a cell membrane that allows some things to cross through it and not other things. And that is called selective permeability. Ability to allow some materials to pass through and not other materials. And this is a very important function of the cell membrane. And so we're going to try to understand this process in a lab setting. All right, so here's a close-up of the cell membrane with its lipid barrier, its hydrophobic barrier from the fatty acids of the phospholipids. Some things can pass through, small things, oxygen, carbon dioxide, small non-polar molecules, lipids, water. Other things cannot pass through. Other things such as large molecules, glucose for instance, big things. Ions, charged ions cannot pass through. And many other polar water-soluble molecules cannot cross that barrier. So when things pass through it, all on their own, going from high concentration to low, that is passive transport. So going from high concentration to low concentration, that is passive transport. And the cell does not need to supply energy. No ATP, no energy is required. So that is pretty nice. Again, things can move in either direction across the membrane. They just need to go from high to low. All right, so some types of passive transport. We have simple diffusion. So simple diffusion, so often called just diffusion, is anything going from high concentration to low concentration. Osmosis is simple diffusion of water. So osmosis only involves water, the movement of water from high water amounts to low water amounts. And then there's also filtration and facilitated diffusion, but we're not going to talk about those too much in this lab. All right, so here's some examples of passive transport involving diffusion. So you have a lot of dye on one side of a membrane, none on the other side. So which direction will the dye move? Well, it will move toward the low. So high to low is how the dye molecules will move. And eventually, if all is at all possible, it will reach equilibrium where the amount of dye, concentration of dye on both sides is the same. All right, we can also have multiple things crossing the membrane at the same time, but they will move in relation to their own concentration. So blue on one side, red on the other. Blue goes to the right, and red will go to the left based on good old diffusion, high concentration blue to low, high concentration red to low, until in theory equilibrium is reached. And in our bodies, this occurs, say, in our lungs, when oxygen and carbon dioxide are moving either into or out of the bloodstream. All right, which direction will glucose move? Here's the membrane, 10% glucose solution on side A, 1% on side B. Which direction will glucose move? It'll move to side B, going from high concentration to low concentration. Which direction will water move? Water move to side A, going from high concentration to low concentration. With or without membranes, that's how the movement occurs if the movement is at all possible. All right, things that affect the rate of diffusion. Temperature. Higher the temperature, the faster the diffusion. Higher temperature, faster diffusion. Another thing that can affect it is the size of the molecule. Lower weight molecules, smaller molecules move faster. So smaller is faster. Like a 2-year-old through a crowd of people, way faster than their parent. And then steeper concentration gradients. So the bigger the difference between the two sides, between the high and the low, the faster the rate of diffusion. So you can sort of think of it as a steep line versus a less steep line. This gradient, only difference of 10, smaller gradient. This gradient, difference of 70, much steeper. So that is going to be faster. All right, here's some more examples. Here is dialysis tubing with 40% sugar solution surrounded by water. Here's another dialysis bag with 80% sugar solution in a water, pure water, and the holes in the dialysis bag are small enough to allow both water and sugar to pass through. So which of these is going to go faster? Which has the steeper gradient? I hope you said the 80% solution versus pure water has the steeper gradient. So water is going to move in much faster than it would in the other example. Again, bigger difference. All right, so water moves through membranes. Water can pass through permeable membranes, going again from high concentration water to low concentration of water. So the more solute, the less water. So that means 4% protein versus 8%, more water, less water. Water is going to cross over to the A side. You can also think of it in terms of the solutions. Water always goes to the hypertonic solution. So the solution with more solute is the hypertonic solution. Water is going to go to the hypertonic solution. And eventually, over time, if allowed, it will try to reach equilibrium. All right, here's another example. So here we have this tube with a membrane separating a 10% sugar solution from a 4% sugar solution. Comparing the two solutions, the 10% or B is hypertonic, the 4% or A is hypotonic, more solute, less solute. So which direction will water move? It will move to the B solution, to the hypertonic solution. Water will cross the membrane and go to the hypertonic solution. And if allowed, will reach equilibrium when both solutions on either side are isotonic, have the same amount of solute, relatively speaking. All right, some more practice with the terms isotonic, hypotonic, and hypertonic. So solution B here is what compared to solution A? The little stars are the solutes. Solution B is what to solution A? And solution B is what to solution C? Well, I hope you came up with these answers. B is hypotonic compared to A. It has less solute. B is hypertonic compared to C because it has more solute compared to C. So again, these terms are relative because you're comparing solutions. All right, here's some more practice. Here's a dialysis bag in pure water, 5% glucose. Dialysis bag filled with pure water. The holes in the bag let glucose and water both pass through. The fluid in the bag is what to the fluid outside? Fluid in the bag is hypertonic. It has more solute, 5% versus 0. And which direction will water move? Water will move into the bag. So the net movement is into the bag. A little move out, but net movement into the bag, going from high amounts of water to low amounts of water, or going to the hypertonic solution. All right, here's another example. We got 5% glucose solution in the bag, 4% glucose solution in the beaker. The fluid in the bag is what compared to outside of the bag? It is hypertonic still, 5 to 4. 5 is a bigger number, so still hypertonic. Which direction will water move? It will move into the bag. Again, technically water moves both ways, but the net movement is into the bag, going to the hypertonic solution. All right, real cells, lovely red blood cells. When red blood cells are in an isotonic solution, water leaving and water going into the cell and water leaving the cell are both the same rate. So the red blood cell is in its happy place. It wants to be in an isotonic solution. If the red blood cell is in a hypotonic solution, a dilute solution, then more water will enter than leave, and the cell will swell and eventually lyse or burst, and that kills it. And then if you put a red blood cell in a hypertonic solution into a very concentrated solution, the red blood will water will be leaving the red blood cell, and that'll cause it to shrivel or crenate. So here is some pictures of red blood cells that have undergone crenation, have shriveled up. All right, so the solution in an IV bag that gets injected directly to someone's bloodstream, it is blank to blood plasma in the person's bloodstream. So what is it? Isotonic, hypotonic, or hypertonic? Well, I hope you said isotonic. That's what we want. We want the concentration of the IV solution to match the concentration of the blood plasma so that the red blood cells remain in a lovely isotonic environment. All right, other things can't cross the membrane by themselves, but we might want them to, such as glucose, large molecules, ions. How do we get those into or out of the cell? Well, I'm not going to talk about it, but as you know, facilitated diffusion with a protein that lets them go through, yeah, that's the main way for this case, facilitated diffusion. All right, that's it for this.